mRNA LNPs are in a way artificial viruses, and they are perfectly suited to trigger an antiviral immune response if given intramuscularly, and that drives then a very high and good immune response that can be used then for prophylactic vaccine purposes, but also for inducing anti-cancer responses.
Yeah, I mean, really, through the success we've seen during the pandemic, I think the most important part of these mRNA vaccines is really speed to market.
RNA therapeutics as a vaccine platform allows to generate very rapidly these types of vaccines and to test them. It's a fully programmable modality, and therefore this is the reason why we saw these vaccines emerging so fast when the epidemic started. It's just a fantastic modality that you can prototype things, whereas in other types of platforms of vaccines, it takes much more time to tune this type of molecules, and also the production is harder.
One of the advantage RNA and LNP has is it can enhance the expression for a long time, and that's one of the key for the therapeutics.
Now we're not talking about a virus that is live or attenuated or even dead. We're talking about we're taking a subsection of that virus and actually a software, or basically the sequence, that will produce those kind of vaccines that will elicit some sort of a response.
So one of the biggest challenges in the field of LNP is to selectively deliver into the disease organ. So basically, I am an oncologist, so in the cancer, in the tumor, and I will say still there are like hindrance how we can deliver into the tumor.
Some of the issues with LNP-based formulation obviously is delivery and making sure that you get sufficient quantities of your RNA to the target tissues of interest. And so I think that's one of the major limitations, and we look forward to the meeting and seeing how there is advances coming down the pipeline.
We've made great strides in delivering our medicines to various cell types and organ systems across the body. We're exploiting all that, but there's still more work to be done. And I think one of the biggest promising areas is targeted delivery, ligand-conjugated antisense or siRNA delivery to specific organ systems like skeletal muscle, cardiac myocytes, endothelial cells in the lung, and other tissues as well. Making great progress there, and the future is really promising.
The biggest hurdle is still delivery into the organ of the interest, and for as an oncologist, delivering into the tumor, that is the biggest hurdle, how we can target LNP to the organ. And second biggest hurdle again means it's kind of still the synthesis of the nanoparticle and optimizing the particle purity.
During this conference, we've been hearing a lot about delivery. So is it going to be LNPs the future? Is it going to be a different kind of conjugation? That you're conjugating your mRNA to a different small molecule? Or is it going to be exosomes? These are really to be determined, and we'll see how this industry really folds within the next few years.
Part of the challenge is that duration of effect. Right now, these modalities are really good, but they have a short duration of effect of only a few days. So you need to keep injecting them over and over again if it was a regular therapy. What we want to see basically in the future is the ability to go and extend their half-life to a much longer time period, and also to deliver them to various types of organs beyond the current ones that are in the clinic.
So one of the biggest, I will say, challenge right now is scale up. So and with the purity too. So there are two things. So how we can scale up the production of LNPs, and second thing is the purity of the particles.
the LNPs are really the major development that we've seen for mRNA delivery. I think in this conference, most of the talks were focusing on what are those next generation delivery, especially for different tissues and different indications. For the different technologies within LNPs, the most important thing is that you want to see heterogeneity and have a really good homogeneous mixing, as well as having a very high rate of full encapsulated lipid within mRNA versus empty. And how do you actually characterize those?
Every step of the production process is important because it will impact the quality and the final composition of your product, and therefore you want to understand every of these process steps.
Downstream processing is very important for the purity of the particle, so for the size of the particle. So buffer exchange and all those downstream filtration is the key and very important part of the drug development.
So PAT is something that is very well known within the biologics industry. Now it's being implemented more in the cell and gene, but I would say for mRNA, that is really where the next cutting edge development has to occur. You have to understand really what your critical quality attributes are, your CQAs, your CPPs, critical process parameters, and from there you'll be able to design a very well PAT strategy.
Every manufacturing process that you want to scale up and make commercially available, you need a control strategy, and analytical methods are the key component of this. So, but the analytical methods alone does not help you. You need to understand which quality criteria are really predictive for your potency also.
I think one of the key things for the nanoparticles is to really see some of the changes in the composition of those nanoparticles, whether it's able to get into CNS, which we heard quite a bit about today, trying to crack that blood-brain barrier, or find ways of, say, ligand-mediated LNP delivery, again, targeting specific tissues that will help with the delivery of those products. One of the things that I think everyone agrees upon is that delivery is key for almost all medicines, and that there's a lot of efforts both on the investor side as well as the big pharma side to crack this, because that will enable new therapies and new developments going forward.
Yeah, we are living in exciting times. We get so much data out of clinical trials. So many trials are initiated and are reading out in the next few months. And that will teach us backwards what preclinical models can be used to develop such systems, to optimize them, and to better understand them. So far, the field was always concentrating on efficacy to get this first product done. Now we are entering the next level, where we are looking into the usability of the materials. Yeah, as discussed, stability, concentration, dilution of the materials, pre-filled syringes. That will open up the next stage of this technology.
So I think one thing that we're going to see in the field of nanoparticles is we're going to see a more rationalized design, because we're going to screen in a high throughput manner a large number of nanoparticles and to understand the structure-activity relationship between these nanoparticles and their ability to enter into specific cells and to introduce the bioavailability of these molecules. So that's, I think, the biggest revolution in the next 5 to 10 years.
There's a lot of emerging great RNA science happening, and I'm really excited to be part of it.